Okay, so now I'm going to talk about QC, quality control steps. So once you've created your single cell experiment object, so you have your expression matrix, you would like to check whether you have outlier cells or outlier genes that you want to remove from your data and not use in the further downstream analysis. So here we will use a data set. We call it the Tang data set. Tang is the first name of the first author of the paper. Why we chose this data set is because this data set has several batches. So the data comes from 3 different individuals, and in each individual we have 3 batches. So we wanted to show you some kind of real data set, but the principles that we use here and the functions you can apply to any other data set. And as Davis mentioned in the previous chapter, we're going to use single cell experiment object and scatter package for doing quality control. So also the advantage of this data set is that the authors provided both molecules counts and read counts. So I will cover the quality control of the UMI molecules, and in the next chapter you will go through the read quality control yourself, and we'll basically just repeat this analysis for the reads. And then you can compare how molecules are different from the reads. So we'll start with loading the data sets. So we load the molecules. We also load annotation data for the cells. If we look at these matrices, the molecules data looks like this. So you have cell ID in columns and gene ID in rows. And here we have UMI counts from your single cells. And the cell IDs have 3 parts. So the first part is individual ID, the second one is the replicate ID, and the third one is the well ID. So if you look at the annotation matrix, annotation matrix describes your cells. So for each cell ID, this is your cell ID in the expression matrix, we have several columns describing what this ID means. So now we create a single cell experiment object. Because we have counts, we assign, we create a counts assay in our single cell experiment object, and we say that the cells are annotated with the annotation matrix. Then we'll do some simple quality control steps. So the first one is we can just remove all the genes that are not expressed in any cells. Those genes are not informative, and we don't need them at all in our analysis. So this line will create a logical vector called keep feature. And then, as Davis mentioned before, we can easily subset our single cell experiment object using the logical vector. So we say we want to remove the genes. We want to only keep the genes which satisfy this criteria, and this will subset the genes and also subset the annotation for the genes, but we don't have annotation for the genes. But in your own experiment, when you have it, everything will be done automatically here, so you don't need to worry about separately subsetting your matrix and annotation.
So this is here you are using the unique molecular identifier.
Yes.
As that's your key to get the expressed genes, is it right?
Could you repeat the question?
You are using the UMI as your key to get the expressed genes or the expressed cells.
So the question is, we're using UMI as
UMI is the first step, okay, for the genes which has been expressed.
So UMI here is a single cell experiment object, which contains contains our expression matrix in the counts assay slot of the object. All right, so UMI is the single cell experiment object, but inside of it you have a matrix of UMI counts from your experiment. That's why we call it UMI.
But to my understanding from the beginning, from the introduction, the UMI would be like a particle would be.
Okay. Okay, so the question is whether this UMI is the same as UMI that we had in the previous chapter. So the name is the same. We call it UMI. The only thing, the only reason why we chose UMI name here is because in the next chapter we're going to work with the reads, and we will call that object reads instead of UMI, so you can distinguish between them. You can call this object as you want. It doesn't matter. But yes, so this matrix is UMI counts, and that's why we call the object UMI. It's just the name is the same, but yeah. Does it answer your question? So in this matrix, we also have some genes which start with ERCC. These genes are spikings. So in some of the experiment, you would have, if you added your spikings to your experiment, you would know that. And then you will have genes with ERCC names. So in this experiment, we have genes with ERCC names, and that's why we want to define them as spiking. So there is a function called isSpike, which you apply on your single cell experiment. And then you call it ERCC, this new slot in your data set. And then you just select all the genes which start with ERCC in the beginning. We also know some of the genes which are mitochondrial genes. And these genes are usually also very useful in the quality control. So if you look at the expression of mitochondrial genes, you can see whether whether the cells are dying. Then the expression of these genes will go up. So we also define mitochondrial slot in our object and provide manually the gene names for that. So usually, I mean, this is an optional step, but usually it's a good idea to look at mitochondrial genes. So here we define them manually. I think these genes are taken from the publication of the tank data. So in real life, I'm not sure. Talul, do you know, for the mitochondrial genes, is there any database that you can use to define them?
They'll be in the GTF, your genome annotation.
Okay, yeah, so they would be defined in ensemble. So if you have aligned
also,
your...
if you annotate with biomark, then you can find which are on the mitochondrial chromosome.
Yeah, I mean, there's a very small number of mitochondrial genes, so it should be quite easy to find based on the names as well.
Yeah, probably you can use this one for human. This is a good set of mitochondrial genes. And then we run calculate QC matrix function on our object. And in this function, we can also define what our feature controls are. So we say that we have two controls, ERCCs and mitochondrial. And ERCCs are contained here, and mitochondrial genes are contained here. So this will run quality control on your data set, keeping in mind that we have ERCCs and mitochondrial genes. So when we run this function, this will create additional columns in your annotation matrices. For example, if we had a cell annotation matrix with names of the patients, names of wells, this function will add some extra columns to this matrix, which will contain information, summarized information about the expression matrix. So if we look, for example, one of the columns would be total counts. So that function will calculate total counts per cell and will store it in the annotation matrix of your data set. And you can access it by using $sign. So here we would like to plot the histogram of total counts in each cell in our data set. And we can see we have this kind of distribution. So you have some cells which contain 200,000 counts. Some cells are very close to 0. So this already gives you some idea of whether your experiment was good, of good quality or not. Some cells which are probably outliers, some cells are something strange happened to them. So this distribution gives you the first idea of whether your experiment is good. So we have the first exercise here. So when you look at this plot, you can already, as I said, find outliers and you can remove some of them. So here we removed, we put a threshold at this position because these cells are very close to 0. So there are only a few molecules in them. So we don't want to use them for further analysis. So we have two questions here. How many cells does our filter remove? And what distribution do you expect that the total number of molecules for each cell should follow? Okay, so actually we forgot to provide the filter expression here. As I mentioned before, you can go to the RMD file in the RStudio and find out solution to this exercise. Well, actually, sorry, I'm wrong. So here, when we plot this red line, we define the threshold of 25,000 total counts. So our filter will remove all cells that contain less than 25,000 counts, and we'll keep everything else. And if we apply this filter, this will remove 46 cells and will leave around 800 cells in the data set. So this is our first filter. And if we come back to the question, what shape of this distribution is good or not, what are your thoughts? What would you remove from this data set looking at this distribution? Do you have any ideas? Yeah, so that's a very good idea. So the idea is that sometimes you might have two cells basically connected to each other and they are not dissociated. And that's why when you sequence those two cells, you will have twice as many molecule counts as in other cells. And that's why maybe these cells are actually doublets. These cells are called doublets. So when you have two cells in the same well, for example. So yeah, so that's one very good idea. So if you have cells which are very highly expressed, most probably something is not good in the experimental conditions. Those cells that are very lowly expressed, the same. Something went wrong. But we think that the perfect shape of the distribution obviously would be a Gaussian, right? And if it's located somewhere here, that would be a perfect experiment. But you always have cells which are lowly expressed, cells which are highly expressed. So at this point, you have to decide yourself which cells to exclude. It's very hard to do it in an automatic way. We can do the same exercise with the number of expressed genes. So I was asked at the break, I was asked what are the expressed genes. So in contrast to the previous histogram, in the previous histogram we plotted the sum of all molecule counts per cell. Here we plot a number of genes that are expressed in each cell. Okay, so instead of looking at the original matrix, we create a binary matrix from it, where we have zeros and ones. 1 means that gene is expressed, 0 means that gene is not expressed, and then we count the ones in each cell. And this is the histogram of these values. So this looks actually better than the previous plot. So you have a Gaussian which is not close to 0. But here we can see that some cells contain very low number of expressed genes. So probably, again, we should remove them because they don't follow the general trend in your experiment. So here we apply a filter of 7,000 genes, which is a good number. So if you have less than 7,000 genes expressed in the human genome, maybe something went well. It's related to this experiment. So in your own experiment, the value will be different, obviously, and also depending on the protocol. So some protocols are deeply sequenced, some protocols are not. Yes.
Essentially, like how do we set the threshold of where to cut it off? Like how did you come up with the 2,500, or on the other end?
So the question is how do we set the threshold? So here we set the threshold manually by looking at the histogram.
Oh, you just go like.
Yes. So it's completely subjective, and we think there is no other way to do it at this point.
So when you build, you said you have a matrix with ones and zeros according to if it's expressed or not. So how do you define that a gene is expressed? Is there a threshold of the number of genes that come from that?
The gene is expressed if its value of molecule counts is more than 0.
Just zero.
Yeah. You can define a threshold for your genes. So you say, I want my genes, I call my genes expressed if it contains more than 5 molecules. This would be a manual threshold. But here we just, we go for the easiest one.
But is there any normalization for the length of the gene, for example? Like when you decide if a gene is expressed or not. And what's the advantage of using one or the other one? Because if I use the UMI, if I use the read-based threshold, or use the gene threshold, what's the advantage of one over the other?
So the question is, are there any normalizations for genes which consider the gene length, take into account the gene length, and also what's the advantage of using total counts versus total features? So there are normalizations for gene lengths. We'll consider this in the next chapters when we look at the normalizations. Advantage of using this one against the previous one, there is no advantage. I think we should use both, because these are the first 2 main features that you can look at in your data set. So it gives you the idea of your data set. Any other questions? So here we have exercise 2, which is related to this histogram. So yes, here we have some comments which say that, again, as I said, these histograms vary depending on the protocol. So in droplet-based methods, you will have lower sequencing depth, and so fewer genes will be expressed per cell. So this distribution will be shifted to the left. So you have to be careful and understand your protocol and thinking what could go wrong in the protocol before you remove the cells. But still, if majority of your cells are in some position here, it's still better to remove the outliers, because you don't want to look at something which is suspicious. You want to look at the majority of the cells. So the gene filter, the filter of cells based on the number of expressed genes removed 116 cells and kept 748 cells. So now we will use our controls, spikings and mitochondrial genes, also to look at the data and see whether our controls give us some more information about the quality of the cells. We will use this function. This is a scatter function called plot pheno-data. So you can provide some arguments to the function, and these arguments that we use here mean that we want to plot pheno-data, have total features on the x-axis. We want to have percentage of counts in mitochondrial genes in y-axis, and we want to color the data by batch. So batch, all these things are columns of our annotation metrics. As I mentioned before, when you run quality control function, it will create additional columns in your data. So these are columns of the annotation metrics. And in Scatter, we can actually, using this one function, you can plot a very nice plot of your data. So here we look at the mitochondrial genes. So total features means number of expressed genes in your cells. PCT counts, mitochondrial means percentage of counts in mitochondrial genes. So this plot shows us that majority of the cells have only 5% of counts in their mitochondrial genes. So if you look at the mitochondrial genes, sum up all the counts in mitochondrial genes and divide them by the total number of counts in the cell, this will be 5%, right? But there are some cells which are quite high. So these two cells, for example, contain more than 15% of counts in mitochondrial genes. And if you remember, the number of mitochondrial genes that we defined was like 10, right? So 10 genes, but they contain 15% of counts. So, sorry? No? So something could go wrong in those cells, right? So we want to remove them. So that's how we evaluate the dataset quality, by looking at mitochondrial genes. And then we can do the same with ERCCs. So instead of MT here, we call ERCC. So these are your spikings. And here we have really interesting plot, because most of the batches contain reasonable number of counts in their spikings. But there is one batch, this one, the orange one, where all the ERCCs are much higher than in the other batches. So this is an indication that this batch is different, right? So probably we should remove it. And we will remove it afterwards. We will not consider it. So sometimes something can happen with one batch, and it will be different from others. So we don't want to look at it in the downstream analysis. Yep.
With the mitochondrial reads, are you more concerned about a trend or about an absolute number? Like, are you always going to cut off anything above 5%, or what if everything was around 10%?
It depends. Yeah, the question is, in the previous plot, we didn't see any differences between batches, so all the batches were mixed. So we define a threshold somehow manually. So the question is, shall we do it every time like that or not? So I think, again, it depends on the experiment. So sometimes this could go up to 80%. So it's very hard to say. You could have a batch effect on this plot as well. So it's just in this experiment, the batch effect we can see in the spikings, but not in mitochondrial genes. But it's not always the case. So it's always, again, these two plots, I would say it's always a manual selection of cells that you want to remove. And the advice is remove some outliers, right? Keep the majority cells in the data set, remove the outliers.
But it does seem that there is one batch that has the high mitochondrial counts, right?
Which one?
I mean, if you look at the color, isn't it the replicate 3 of 19101?
This one?
Yes.
So the question is, it looks like this replicate has a batch effect, which I don't really see on this plot. Can anyone see any batch effect with this replicate? Is it any different from others? I don't see it. If like
All the cells above, like 12 for the ECT count, are on that edge.
okay. All right. Yes. So yes, there is some batch effect here. Yes. But still, you still have some cells here, right? So we just remove a few cells instead of the whole batch. Because like this is not a very strong indication that the whole batch is wrong. Whereas if you look at ERCCs, definitely here it says that the whole batch is wrong. Well, you can actually keep it in your data set if you want to, because it looks like it's not completely out of space. It's somehow like scaled, right? So when you do normalization, you can normalize it back to the level of other cells, I suppose. But we don't recommend that, so it's better to remove it. Yeah. Yes, this is all. Yeah, so the question is, because usually we have around 100 ERCCs in our data set, here we plot the sum of all the ERCCs in your cells. The question is whether it's worth plotting each ERCC separately and look at them individually. You could do that, but then you'll have 100 plots, and it will be very hard to analyze. And but then, okay, yeah, but then when, let's say you have two spikings in two plots. In one plot, your cell is an outlier. In the other plot, your cell is not. What would you do with this cell? That will be complicated. So I think it's very simple and straightforward, right? So it's for quality control, that's fine. We will actually look at, Talula, are we considering ERCC normalization in the normalization chapter?
Yes.
Yeah, so we'll look at normalizing your data by using ERCCs where you take them individually instead of the sum of them. So our mitochondrial filter removed 96 cells, and ERCC filter, ERCC filter removed 96. Actually, you can see this was the number of cells on your plate. So we removed the whole batch. Usually it's 96 cells that you have in this experiment on your plate, whereas mitochondrial filter removed 31 cells. So here we have another exercise which asks about the size of the cells. What would you expect to see in the RCC plot if you were examining a data set containing cells of different sizes? So we provide an answer. So you would expect to see a group corresponding to the smaller cells with a higher fraction of ERCCs and a separate group corresponding to larger cells. And the reason for that is that you add the same amount of ERCCs into smaller cells and into bigger cells. So the concentration of ERCCs in smaller cells will be higher. All right? And sometimes this is very critical experimental condition. So sometimes you're actually interested in the sizes of the cells. So you have to keep in mind how large are populations of your cells in the experiment. In most of the experiments, people just look at all cells and assume that the size is the same, but sometimes the size makes a difference. Okay, so now once we have defined all these logical vectors for which filter out cells, some cell outliers, and we can assign this combination of these all 4 filters to the use column of the annotation matrix. So we just combine these logical vectors with AND statement and put them into the use column of the annotation matrix. And the combined vector contains 200 cells removed from the dataset. So we can then use this filter at the end once we've done everything. So basically by looking at the data manually, using 4 different plots, we were able to remove 200 cells out of 800 cells from our experiment. Sometimes you don't want to do that, so sometimes you have small number of cells and you don't want to remove half of them, right? But if they're bad, you have to do it. Though otherwise, the downstream analysis won't make any sense. So Skater also provides some automatic ways to detect outlier cells in your matrix. So this is based on several features. So Skater, if you run this function, plot PCA on Skater object, on single cell experiment object, and you ask it to find the outliers in your data, it will look at these features, at these columns in your annotation matrix. All these columns were created by quality control function, calculate QC function. So Davis thought that these features are the most important ones for finding cell outliers. So when doing the PCA dimensionality reduction, it will use these features. And then a PCA plot based on those features will look like that. And Skater can also find, like, in an unsupervised manner, find outliers in this plot. And here they are colored in orange. So Skater basically find that you have lots of cells are good cells. Then there are these cells. Probably it should take these cells into account as well, but the algorithm only defined those ones. So if you don't want to do that manually, all these plots that we plotted and looked at the data, you can run this function, and Skater will do it for you. But this is all unsupervised, so you cannot be sure what happened here. Yeah. Yeah.
Does it really matter? I mean, how does it matter? Because then that suggests that even though the amount of RNA is low, the number of transcripts was high enough to be similar to the cells. You see what I mean?
Yeah. So the question is, as I understand it, why would we do so many QC steps?
No.
Right?
So in the ERCC, you're checking for endogenous RNA.
Yes.
As QC. So these cells in sort of orange. The idea is that because the ERCC is so high, the endogenous RNA is low.
Yes.
But then those cells were filtered out at the beginning when we looked at the amount of transcripts per cell.
Yeah.
So this suggests that even though the original amount of RNA or cDNA sequence was low, the number of transcripts for those cells was good enough to pass the beginning of the QC.
So the question,
Matter.
yeah. So the question is, in the first plot we had total number of counts per cell, the histogram of total number of counts per cell, and we probably didn't remove these cells by using filtering on this plot. Whereas in this plot we can see that there is a batch which contains cells where percentage of counts in ERCCs is much higher than in the rest, right? So this means that, for example, this cell and this cell can have the same number of total counts, right? But in this cell, only 5% of these total counts belong to ERCCs, whereas in this cell, 15% of the total counts belong to ERCCs. And that's why we want to filter these cells out, all right?
So the beginning takes into account
In the beginning, we don't look at the ERCCs, we look at the whole thing in general. Here we look specifically for ERCCs. All right. So if we run automatic outlier finding in Skater, Skater found 45 cells that it wants to remove, which is much lower than what we found manually. Yes.
What PCA, if it tells you that the number of features that it pointing to one of the dimensions or one of the components, then you can kind of roll it back to know whether it is basically filtering based on like ERCC genes or is it filtering based on different features? Is it possible to kind of like interpret it reversely?
Yes. So the question is, in this PCA plot, we can always, this is PC1, this is PC2, we can always go back to the PC1 and see what is the main component of PC1. So this PCA plot is based on these columns of the annotation matrix, right? So we can always find out which of these columns had the most effect on PC1. This is called, how is it called? Loadings, yes. It's called loadings on in the PCA. So yeah, it's possible to do. So you can, I don't know whether Skater writes this dimensionality reduction matrix into dimensionality reduction slot.
Not unless you explicitly tell it to.
Okay, so you can define a parameter here and say that you want to write the results of this to dim-rad slot of the single cell experiment object, and then you can explore those PC's components in the slot. Yeah.
Are the spikers added before amplification or after?
The spikings, are they added before amplification or after? They're added before. Yes.
Usually during reverse transcription.
Sorry?
Usually just before reverse transcription.
Before reverse transcription. So if we come,
sorry, but it should be added that the spike-ins are sort of derived from bacterial sequences. They're very short and they don't have poly-A tails, so hence it's believed that they behave quite differently from the endogenous RNAs. So that's important to keep in mind.
so if we compare the filterings, we will see that our manual filtering also picked up all these 45 cells that Skater picked up automatically, but we also have 162 cells in addition. Okay, any questions for the cell filtering? Is it clear? Yep.
So, is there any need to use the spikings really? Because if you can use the by transcript command, and if you don't have the spikings at the beginning, then you could just exclude your cells by the expression level of your cells without having the spiking anywhere to start with. So say you have so if you sequence your data, then you have that most of your sequences contain spikings.
So
And if you can do it really, if you can proceed without having them in, I mean, is there any particular reason why, apart from knowing, so it seems,
No.
If you can detect that by local transcripts anyway,
No, I think that the purpose, sorry, the question is what's the point of using spikings if you can use some other filtering procedures? So I think the whole purpose of using spikings is that the assumption is that you would have the same amount of spikings in all the cells if the experiment was good, because those are bacterial sequences, right? So you should have the same amount of spikings in every cell. And then if something went wrong in one of the cells, then you will see this in the spikings as well. And then you can remove it, the cell from your experiment. So the whole purpose of spikings is they are like indicators of quality. Another problem with spikings is that sometimes, because you add them manually, sometimes you could add more into some cells and less into other cells. So I heard a lot of people complaining about it. And in that case, different amounts of spikings will not correspond to the real quality of the cell. It will correspond to the quality of you pipetting them into cells. So I think this question is very personal. Some people like spiking, some people don't like spikings. But if the experiment is perfect and it works, then spikings have to be used, because they will tell you which cells are wrong. All right, so if there are no more questions about cell filtering, we'll move on to gene filtering. So once we looked at the cells, now we can also look at the genes. So there is a plot QC function in Skater. If you run it with this argument, it will show you the top 50 expressed genes in your data set. And as we can see, some of these genes are actually spikings, they probably come from the batch that we excluded, right? Because they were very highly expressed here. So it gives you an idea. Like we don't know the names of the genes because we used ensemble IDs in the beginning. But if you use gene names when creating the single cell experiment object, you would immediately see some genes that you probably know about, some genes which shouldn't be here. So it will give you some idea of what happened in the experiment. So now we would like to remove genes whose expression is considered undetectable. So before that, I was talking about 0 threshold on molecule counts. So if you think that your threshold should be a bit higher, you can define it yourself. So here we say that we want to remove genes. So what do we say here? Define a gene as detectable if at least 2 cells contain more than 1 transcript from this gene. So we say if there are at least 2 cells with at least 1 transcript for this gene, we will keep this gene. If it's less, if it's only 1 cell, then we'll remove it. So you can define these thresholds yourself. What's the good way of doing it? It's hard to say, but usually 0 is probably not a good threshold. So I agree with you, 0 should not be used as a threshold, but something like, if you know the sequencing depths of your protocol, if you look at the total counts per cell, you can have some idea what would be your minimum threshold. And then you can also define what's the percentage of cell that the gene should be expressed in to keep it, right? Yeah. Filter before normalization. The question is, why do we filter before normalization? So we think that these genes are not informative. If they're not really expressed, why would we keep them in the matrix, right? They will not make any difference in the further downstream analysis. They will not make any difference in normalization as well, because most of them will contain zeros. Well, that's what I'm saying. If, say, your gene is expressed in 5% of your cells and very lowly expressed in 5% of your cells, most probably there is nothing interesting there.
So one of the reasons that people they do single cells because they want to look at the lowly abundant genes that they would use if they do bulk RNA.
So
Very biologically interesting.
So you're saying that people do single-cell RNA-seq to find lowly expressed genes which they cannot detect in the bulk experiment, which is probably I think it's a very specific question that you can ask. Then you wouldn't remove it.
No, you can't. That doesn't make sense to do that. Generally, you have poor detection rate in single-cell RNA-seq, so you detect fewer genes overall. So a good single-cell RNA-seq, you're getting about 6,000 genes per cell, which is a lot lower than you would get from a bulk RNA-seq experiment.
Those detection limits we discussed earlier today, like there are, depending on the technology you're using, but generally speaking across single-cell technologies, it's difficult to detect lowly expressed genes. So I would think very carefully about the experimental design before doing a single-cell experiment where it was crucial to be picking up lowly expressed genes.
Yeah, I mean, unless you're sort of, the reason is that the gene is lowly expressed in your bulk because the cell type is rare, but if you know that it's actually or suspect that it could be highly expressed in a low number of cells, then of course it would be meaningful to do it for that reason. But then it's no longer lowly expressed by definition.
Yeah. So if the gene is expressed in 5% of the cells but highly expressed, then it makes sense. If it's very lowly expressed, it's not. It's probably an error in the expression. So here we define this filter. So we want at least 2 cells with the value of gene expression more than 1. Then we will keep this gene. If not, we'll remove it. Yeah.
I mean, is there a way to set a threshold that is cell-specific that depends, say, on the spike ends, the expression of the spike ends?
So the question is it possible to set a threshold which depends on spiking's level? I think it is possible. Obviously, I'm not sure whether it's meaningful, but.
Because I mean, then in a heterogeneous cell population, you would think that this would make sense because then if one cell.
But I think the whole point of spiking is that you expect them to be the same in all cells, right? So your threshold will not depend on spiking level. Should not depend on spiking level. So this gene filter removes 4,600 genes approximately, which is fine. So we still have 14,000 genes we can work with. So as Tallulah said, usually you have less than 10,000 genes to work with. So I think 10,000 is a good number. A bit less is a good number. So the more you remove, the better. So now we want to save the data. And now we have, because we defined these filters now in the raw data matrix, which is an annotation for genes, and cold data matrix, which is annotation for cells. We have these 2 filters, so we can just subset our single-cell experiment object by these 2 filters easily. And this is the final dimension of our expression matrix. So now we have only these genes, 14,000 genes, and 650 cells after filtering both genes and cells. But still, our single-cell experiment object only contains counts, assay. So we would also like to log transform the counts and write it to another assay called log counts raw. We will need it for the next chapter. We just need to do it now. This one is, so after we've done the PCA, I think it actually recorded the results of PCA to the reduced dimension slot. So we want to make it null as well because we'll be using other PCAs in the next chapter. And then we can save it to this file on your hard drive. So log normalization we'll cover it in the next chapter, but usually you have to do it for your single cell data to be able to work with your data in the downstream analysis, because working with raw counts is very risky, because you have completely different scales. Some counts will be very high, some counts will be very low. So if you log normalize your data, it kind of scale everything to the same scale. Any questions? So the question is, we removed 25% of the cells from our data set. Is it a good number, or it has to be something smaller or bigger? In my experience, this is a good number. Sometimes you would remove half of your cells, sometimes none of your cells. So it really depends on the experiment, right?
Yeah, it often depends on how easily dissociated your cells are. So if your cells are quite happy being dissociated, you would expect to be throwing out fewer cells. But if it's a complex tissue that's hard to dissociate, you'll generally be throwing out more. Yep.
So if you use the published data and you find that 25% of cells are bad, then it shouldn't be published then, right? Yeah, so, well, it shouldn't happen if you use public data. Usually public data is filtered, and if you read the methods, you would find that what's provided in the paper is actually already maybe 80 or 70% of the original data set. Okay, so if there are no questions, then you can move to the next chapter and do the same exercise. All the steps that we've done for molecules, UMI counts, you can do them for the reads instead. And look at the distributions at RCC plots, mitochondrial genes, and see what the differences are between the reads and the UMI counts. So we have until 12:30, right? So you can work on that until 12:30, 15 minutes. Then we'll have lunch for 1 hour until 1:30. Then you will be able to continue with the reads a bit longer. Then we'll answer the questions and continue with the next chapter.